me introduce our second speaker, and Nate Traaseth. So it's good that Marina talked a lot about biophysical insight from her techniques, and Nate's work is also of that spirit. So Nate Traaseth did his undergraduate and PhD and postdoctoral work all in Minnesota. He got his PhD in Gianluigi Biglia's lab studying phospholamban and sarcolipin using a combination of solution NMR and oriented membrane solid state NMR, but also at that time, I think he already moved into magic angle spinning NMR. So I guess the only magnetic resonance he hasn't done is EPR. So, and then the work in Gianluigi Biglia's lab went very well, and he continued on to do a postdoc there. Then in 2011, he decided to leave Midwest and move all the way to New York City and started his own lab in New York University, where he decided to move into or study the membrane transporter EMRE as the first system, but now also branching into other transporters. And this is a well-chosen topic because as an anti-parallel dimer that transports in an alternating access way the combination of orientation measurements by oriented membrane NMR and magic angle spinning measurements and looking at conformational dynamics using solution NMR is really just a very potent and well-suited mix of technologies. And so, so he did a lot of that very nice work, but also has started working on the second system, looking at tyrosine kinase, a receptor tyrosine kinase, and here he's interested in mostly, I think, employing solution NMR to understand how hormone binding, in this case, I think it's fibroblast growth factor binding to the extracellular part of this large receptor triggers conformational changes in the intracellular domain, kinase activity, and all that is transmitted through a single transmembrane helix. And so I guess he's going to tell us about using R1 row CPMG type of experiments to look at conformational dynamics and how that underlies this allosteric long range communication and regulation of this protein. So welcome, Nate.
Thank you very much, May, for the introduction. Hopefully everybody can see my screen and hear me all right. Thank you very much for the introduction and for the chance to tell you about what we've been working on. So as May pointed out, I'll tell you about some of our work on the kinase domain. As she pointed out as well, we're also very much interested in transporters. And so I can probably skip this and tell you about introducing the system that, that we're interested in. So receptor tyrosine kinases are really important molecules in biology. There are 58 different RTK genes that are found in humans. These different genes are grouped into different subfamilies of RTKs based on the receptor similarity. And so there are 18 subfamilies that have kind of a prototypical fold where there's a receptor domain connected to an intracellular tyrosine kinase domain through a single pass transmembrane helix. These proteins are very important in cell signaling processes by which they modulate cell behavior in the form of growth, differentiation, survival, and metabolism. Perhaps you've heard of the insulin receptor kinase, and that's certainly a key molecule involved in metabolism. There's also a lot of mutations that happen in receptor tyrosine kinases, and it sort of provides a fruitful avenue for studying and making correlations between structure-function relationships. So the canonical view of how receptor tyrosine kinases are activated in 54 of 58 RTKs, with the one exception being the EGFR subfamily, is through activation loop transphosphorylation. This means that when hormone binds to the extracellular domain, the first intracellular signaling pathway involves phosphorylation in trans. So one kinase has to phosphorylate the other kinase. And that is a critical event in activation because in the unphosphorylated state, the kinase is held in an inactive conformation with the role of A-loop phosphorylation elevating the activity of the kinase to enable downstream substrate phosphorylation. So in my group, we really use pathogenic mutations that are found within the kinase domain now in other places as well, as a tool to try to gauge what are the physiological control mechanisms that hold the kinase in the inactive state. And this is really useful from the perspective of gain-of-function mutations, because the gain-of-function mutations can bypass the need for ligand-activated A-loop tyrosine phosphorylation. So they don't need the phosphorylation to have activity. They so-called have intrinsic kinase activity that bypasses this. So it gives us a good tool to look at the mechanisms of RTK regulation. As May pointed out, in my group, our favorite RTK is the fibroblast growth factor receptor. There's a lot of fruitful biology here. There's 18 different FGF hormones and four members of this subfamily of RTKs. This is showing you one pathway through the MAPK signaling pathway that leads to cell proliferation, but there are also other pathways as well that FGF activates. One of the beauties of studying the system is the large number of mutations that are found in FGF receptors, and several are present in the kinase domain. And these lead to human growth disorders such as dwarfism, as well as a number of human cancers. And I should point out that this project also is a really outstanding collaboration with Musa Mohammadi's group at the NYU School of Medicine. So the first thing that we did when we started the group at NYU was to focus on the effects of one of the clusters of hotspot mutations that are found in the A-loop. And these mutations happen at a lysine residue at 659 that's conserved in all FGF receptor members. And what you find is that these 5 different mutations that happen at this lysine residue really manifest variable phenotypes. So in the worst case scenario, the K659E mutation is neolethal, whereas these progressive other mutations have lower or reduced growth phenotype severities. The interesting perspective from a biochemist viewpoint is that these mutations, which are the only mutations that happen in these sort of dwarfism-like growth phenotypes, is that if you simply purify the kinase domain, you can recapitulate the order of the growth phenotype in a tyrosine kinase activity experiment. So that means is that the K659E, which was neolethal and the most prominent mutation from a clinical perspective, also gives the most tyrosine kinase activity, whereas the least severe phenotype of the K659T mutation gives the least kinase intrinsic activity. So each of these mutations show a gradation between the unphosphorylated wild type, which is the least active, as it should be, and the phosphorylated, the physiological activated. So our collaborators, being good crystallographers, set out to crystallize all of these A-loop mutations, and they were successful, as they have been in many other determination of crystal structures of FGF receptor kinases. But what they found is that each of these A-loop mutations essentially crystallized the kinase with the same A-loop conformation. All were in the active state, and therefore didn't really explain the role of this gradation effect between the severity of the mutations. So, of course, NMR can be a very powerful method in this approach, and this slide is to indicate the quality of the data that we can obtain on the FGF receptor tyrosine kinase domain. On the left is showing you backbone TROSY experiments, and on the right is showing you HMQC methyl labeling experiments using the approach from Gardner, Rosen, and Kay of the precursor additions. So with these high quality spectra, we could obtain really good quality NMR data sets for the different mutations as well as the phosphorylated A-loop. And we can then construct chemical shift perturbation plots relative to the unphosphorylated wild type. And what we see in this construction is that there's really 2 major hubs of chemical shift perturbations. One is found at this kinase hinge region where the other set of mutations, the molecular brake mutations, are located. And the second site is at the catalytic loop. And what you can see is that the phosphorylated wild type, A-loop, shows the highest amount of chemical shift perturbations. And that's followed by this K659E mutation, which was the most severe phenotype and the highest activity in vitro experiments. The threonine mutation showed the least effect. So this kind of recapitulated all of the tyrosine kinase activities. We can look at a specific residue a leucine residue that's found within the catalytic loop of the kinase. We can then color code the spectra of this one peak to the different forms. And what we find is that each of the mutations are located between the wild type unphosphorylated kinase and between the activated phosphorylated kinase. And as many of you know, if you see this kind of linear trajectory of chemical shift changes in the spectrum, that is emblematic of a two-site chemical exchange process that's undergoing, and one in which we can assign those states based off the activity measurements to unphosphorylated and phosphorylated states. Based on the positions of this particular residue, as well as several other residues, we could then go ahead and calculate an NMR-derived population for the amount of active state by assigning the wild type to be 0% active state and the phosphorylated A loop to be 100% activated state. We could then use those NMR-derived populations and correlate those with the activities measured in kinase in vitro experiments. And we see a pretty nice correlation in that the states with the highest NMR populations correspond with those with the kinases with the highest activities. So this suggests a mechanism whereby the gain-of-function mutations work by stabilizing that active state structure, likely by reducing the rate constant of the kinase and going from active state to inhibited state. And this actually makes a lot of sense with the crystal structures when we went back and analyzed these, in that the A-loop contacts that are seen in stabilizing that conformation really have the most intramolecular contacts between the glutamate mutation and the least amount for the threonine. And that makes sense with this, this model where you have a change in the equilibrium constant toward the activated conformation that's enabled by the greater stability of the active state conformation. So then we went on to look at the mutations at this other hotspot location called the molecular break. And the molecular break was coined by our collaborators. And it consists of 3 different residues: an asparagine residue, a glutamate residue, and a lysine residue. And I'm showing you here the inhibited crystal structure and the active crystal structure. And what are seen if you analyze a lot of kinase crystal structures is that in the inhibited states, or presumed inhibited states based on the particular unphosphorylated enzymes that are used, you see that you see really inter-residue hydrogen bonds or salt bridges formed that are broken in the activated state. So we also looked at the bioinformatics of these 3 residues to see if they're shared in other tyrosine kinases. And this is showing you the % conservation as a function of different residues for the 3 different positions of the molecular break. And in fact, you see quite good correlation or good conservation of the glutamate and the lysine residue. The asparagine doesn't show as high of conservation, but that can be explained by the fact that its backbone also can make a hydrogen bond with the glutamate. So in the process of making some of these samples to study these with NMR, we noticed that the molecular break mutations really showed reduced stability, as seen by precipitation, for example, in some of the purifications of the proteins. And so we went ahead and carried out differential scanning calorimetry to get a good quantitative measurement of this stability. And in fact, what we observed were that each of these 3 mutations did show reduced melting temperatures relative to that of wild type. And that kind of explained a little bit of a conundrum in the field, that these molecular break mutations are known as some of the most active mutations if you measure the kinase activity in vitro. But perhaps they don't manifest as strong of phenotypes in patients. They do show developmental developmental disorders, but not to the extent of the A-loop. And perhaps that's because they're not quite as stable in vivo. And this contrasts a lot with what we've observed in A-loop mutations or the phosphorylated A-loop that do not show this reduced melting temperature. So that sort of makes sense that they're using a unique mechanism to decrease stability, and perhaps a unique mechanism for activation. And so we went and did some NMR measurements of the most activating mutant of the molecular break, E565A, and we compared our measurements with those of wild type. And the measurements that we used is a pulse sequence from Tagarinoff and Kay to measure cross-correlated relaxation among methyl group protons. So this gives us an idea for the relative order parameters of those methyl groups. And those are plotted here as a function of residue. This is a little bit difficult to look at, so we made a difference map between wild type minus the E565A mutation. And what you can find are that the change in the order parameter becomes greater, which shows that the relative order of the methyl groups in the E565A goes down, suggesting that this particular mutation increases dynamics. And you can see that a little bit easier in the map on the structure, where in blue are showing you the positions of the molecular brake components, and the heat map is showing you the change in the order parameter. So what we see is these positive, these more orange values clustered in the N lobe of the kinase near these molecular brake components. We also see some quite noticeable changes in order parameters around the phenylalanine of this well-known motif in kinases called the DFG motif, which is just an aspartate, phenylalanine, and glycine sequence in the protein. This DFG motif has been implicated in kinase activation. So because these mutations affected residues around the DFG motif, we went ahead and formed a collaboration with Ying-Kai Xiang's group at NYU to carry out some molecular dynamic simulations. And here we're showing you 3 replicate runs of each, the wild type and the E565A mutation. And we're plotting the Chi1 rotamer of the phenylalanine residue. And what you can see just in looking at this data is that it's flipping much more often in the case of the mutant than it is in the case of the wild type. These particular rotamer angles both correspond with DFG in conformation. So they're both what people in the literature view as active-like states, although we have a slightly different interpretation that I'll share with you in a moment. But we think that the effect of this mutation is then to reduce the hydrogen bond, reduce hydrogen bonding among the molecular brake mutants induced by the mutations. And that actually has an influence on a backbone carbonyl hydrogen bond that we've seen in the molecular dynamics simulation. We think that really weakens or alters the orientation of this side chain rotamer that then has some steric conflict with the phenylalanine, which enables, sort of loosens up that pocket a little bit for the phenylalanine to undergo more rotation. This makes sense with the model that we proposed a few years ago now in 2017 in this eLife paper, where we proposed an allosteric network by analyzing a lot of mutations as well as wild type phosphorylated and unphosphorylated A-loop forms. And what we identified were 4 different sites where there's significant changes between these active and autoinhibited states. And one of those in particular, we defined as called the DFG latch motif. And what happens in the DFG latch motif is that we have a rotation about this Chi1 rotamer of the phenylalanine. And in the case of autoinhibited states, this phenylalanine occupies this orientation, whereas in the activated states, it's more rotated at a different Chi1 angle. So we think the mode of the molecular brakes is to weaken this interaction, which has an effect then on the phenylalanine's orientation, which then can propagate to the A-loop, which is some specifics that were, or some key mechanism that we're still ongoing to try to understand that communication process. So in summary of these 2 particular mutations, we have found that the A-loop mutations really stabilize the activated state in these sort of free energy, schematic free energy diagrams, whereas the molecular brake mutations destabilize the inhibited state. Both have the same effect, which is having a change in the equilibrium toward the activated state. However, they utilize different mechanisms to do this. So the last thing I'll tell you about is a completely different mode of activation that's used by FGF receptor mutations at a different position. And these are found in an arginine residue at position 6, 7, 8. So I'll skip some of the functional data, but I'll tell you that our collaborators and us have figured out that what happens is that this rate of A-loop tyrosine phosphorylation is enhanced by this mutation. In other words, the kinase doesn't possess its intrinsic activity. It still needs the A-loop phosphorylation. However, it can accelerate this reaction. And our collaborators were successful in solving a crystal structure which showed an asymmetric transphosphorylation complex, where one kinase acted as the enzyme acting kinase and the other kinase acted as a substrate acting kinase. So you can see this tyrosine residue right here sort of offers itself into the enzyme's active site and forms kind of what is viewed as a canonical kinase substrate interaction with the catalytic base residue of the enzyme acting kinase. There were additional contexts that were perhaps a bit more interesting to us, which occurred at the C-lobe of the enzyme, and that's blown up on the right here. There's a series of 3 arginine residues from the enzyme acting kinase that made hydrogen bonds or salt bridge with the substrate acting kinase. And this effect of the R678E mutation is seen here in an intermolecular salt bridge that's formed between the glutamate, the mutated glutamate at the 678 position, and a conserved lysine residue on the enzyme. And you can imagine that if that glutamate residue were mutated back to the arginine, as is in the case of the wild type protein, you may introduce some electrostatic conflict between this lysine residue that's on the enzyme and what would be the arginine residue on the substrate. So we set out to try to, 1, validate the effects of the seen in the crystal structure, and 2, gain additional insight into the mechanism of this A-loop transphosphorylation. And the first thing that we did was simply to acquire spectra. These are HMQC spectra zoomed in on the leucines and valine region. We're to acquire spectra at high and low concentrations for both the wild type as well as the mutants at R678 position. So this is the glutamate mutation in the middle and a glycine mutation on the right. And what we've seen with good reproducibility is that the wild type unphosphorylated protein, it doesn't change the homogeneity of the spectra at all as a function of temperature. We can concentrate up to 2 millimolar, and these data are showing you at 1.2 millimolar, where we get very nice homogeneity of the signals. However, the mutations at the R678 position really show attenuations in peak intensity for several residues, both in the E mutation as well as the glycine mutation. And these residues, for example, 667 and 675, are located at the interface of the A-loop transphosphorylation complex. So we think that these data validate that the contacts that are seen in the crystal structure are those that are also seen in solution. Because we had this concentration dependent effect, we went on and tried to carry out estimates of the binding affinity that are present for these particular mutations. And so what we did are use concentration dependent CPMG relaxation dispersion experiments, so multiple quantum relaxation experiments that Louis Kay's group has contributed. And what you can find is that at high concentrations for the residue shown, isoleucine 707, but this is seen for several residues, is that you get very large relaxation dispersions. However, this effect is really diluted out as you go from 1.5 millimolar all the way to 0.1 millimolar. And the same effect is seen in the R678G mutation. So what we could do is fit these curves to data sets obtained at 2 magnetic fields, and then try to correlate these CPMG derived parameters to a dissociation constant. So we worked out this equation shown here, and we can fit our data sets for a few residues with a CPMG dependent parameter shown on the y-axis and the protein concentration shown on the x-axis. And what we're able to obtain are dissociation constants really in the millimolar range, which we think makes sense with, well, 1, the observation from the CPMG that as we go to higher and higher concentrations, we see more and more CPMG relaxation dispersions. But we think this makes sense from the perspective of the wild type as well, where we don't see those concentration dependent measurements, meaning that we believe that the interface needed for the A-loop transphosphorylation complex is really intrinsically weak. And you could imagine Mother Nature has evolved that state to be intrinsically weak to minimize and sort of protect that key event in RTK activation, which is A-loop transphosphorylation. So we think that this makes sense with this model. And if you think about, these are also kinases in solution. But if you imagine kinases at a membrane, that becomes more of a 2-dimensional problem where those binding constants might be effectively quite a bit higher. So we also, because of this asymmetric complex, we went and designed some mixed dimer experiments to gain more insight into this, this binding event that we could observe in solution. So because it's an asymmetric dimer, we postulated that we could make one of the kinases only act as a substrate and one of the kinases only act as the enzyme, because the role of that mutation was really to make the substrate, or make the kinase a better substrate. So if we sort of block this R678E mutation with the second mutation and make a double mutation, we can effectively make it the substrate acting kinase. And indeed, we've shown that in different experiments. What we can use is then differential labeling, where we label only one of the proteins versus we can label the other protein. And these are some experiments that we've done, and I'll show you a subset of those data. We've acquired CPMG relaxation dispersion profiles on the wild type FGFR2, and then that same isotopically labeled wild type mixed in with the double mutation, which will only act as a substrate acting kinase. And in fact, what you see for the wild type only is that they show very little CPMG relaxation dependent parameters, so REX values, as a function of these residues. We see very little for here, very little for here, and nothing for these residues. But that when we add this mutation that acts as the substrate acting, only as the substrate acting kinase, those relaxation dispersion profiles become very large. And that corresponds, again, this validates this interface. But we thought, well, maybe that C-Lobe contact may mediate the enzyme acting kinase to maybe switch toward the active state conformation. However, we didn't see that. We were sort of below the detection limit of these experiments in both cases. And I'm showing you I548 and I623, which we believe are emblematic of the transition from the kinase autoinhibited to the activated state. So to try to gain more insight, we replaced the wild type with this A-loop activating mutant, this K659E mutation, because we know that this K659E mutation does stabilize the A-loops to some extent. So we wondered whether the addition of the substrate acting kinase might increase its active state. And so when we do these measurements, what we find is that in the presence of the substrate, we get the same enhancement on the C-Lobe, which are, again, these two residues located here. But now we start to see increases in the CPMG relaxation dispersion for residues that are emblematic of the transition between autoinhibited to activated state. So we think that this means that the binding at the C-Lobe does have some effect of promoting the enzyme toward the active state, but it's not like a switch-like mechanism where you get 100% active state conformation. It still goes through the kinase's intrinsic dynamics. So the last question I'll summarize pretty quickly here is, we also wondered whether this interface also applies to the wild type receptor. Remember that the mutant was the one that crystallized. So we look back at the crystal structures, and there's these salt bridge interactions between the arginine residues and the aspartate residues. So we figured if we made mutations of these residues, we would be able to then see changes in the wild type's activity relative to these mutations. So no mutations then at the R678 position, only then mutations at this interface, if indeed the wild type shares the same interface. So these are native page gels that were were collected by our collaborators. And so you can see the actual 0P form that you start with and the phosphorylated forms as you migrate toward more negative charge. And these are just Western blot experiments probing for the doubly phosphorylated A-loop conformation. So what you find is that if you make mutations to the two arginines at this interface, you really get very little activation, maybe a little bit after a long period of time. But this really makes sense with this model that if you change these two negatively charged residues, you would create some electrostatic conflict with the aspartate residue of the substrate acting kinase. Similarly, when you make mutations of the aspartate to the arginine residue, you also see a significant reduction in the phosphorylation rate, A-loop phosphorylation rate of the protein. However, when you mix both together, you mix the enzyme acting kinase with this substrate acting kinase, you can recapitulate that activity and you get much greater activity than of the mixture, which is again, we believe, a validation of this asymmetric A-loop transphosphorylation model. Lastly, I just wanted to emphasize that our collaborators also did measurements in cell-based experiments, L6 myoblasts. It's a muscle cell line. And basically, here the same experiments were done with wild type and then these different mutations. And here is showing you minus and plus the effect of the addition of the hormone. It's a pan-FGF receptor activator. And here you're probing Western blots for the A-loop phosphorylation state and then the total amount of receptor present, FGFR2. And what you see is in the wild type is that when you stimulate the cells with hormone for 10 minutes, you get good phosphorylation of the A-loop. However, when you have this double arginine mutation to the glutamates, you see nothing. Similarly, you see nothing with the aspartate to the arginine residue. Again, making sense of that interface that was seen by crystallography and confirmed using NMR measurements. But now when you co-transfect both full-length receptors, now you're able to start seeing some A-loop tyrosine phosphorylation, which is suggestive that the wild type is also using the same mechanism. And in addition to the A-loop tyrosine phosphorylation, we can then also see.
Nate, you should sum up.
Yep. So we also see the activation of the downstream substrate phosphorylation. So basically, this allows us to present the model, and this model is mediated by C-lobes. We also see increased substrate dynamics, and we think that this promotes then the enzyme's active state. So I'll skip this, but it's short to say that we think this is shared in other receptor tyrosine kinases as well. Lastly, I want to thank the people that did the work from my group. Billy Marsiglia, who's a former graduate student, did this work, and he's now a postdoc at Mount Sinai. He was really an outstanding graduate student. And Sissy Zheng really helped Billy with some of the molecular brake mutations. I want to again thank my collaborators and thank the funding, and thank you for your attention.
Thank you. We're somewhat late, but if there, I see one question, and then we'll, or two questions for Nate. I'll just, I think these are relatively short questions. One anonymous attendee asks, are RTK autophosphorylated, or do they require a specific kinase?
So 54 of 58 RTKs undergo autophosphorylation on the activation loop. Usually definitely in trans, normally among the same subfamily will activate. So FGFs will activate themselves, yes. So the answer is yes, most of them.
David Elisa has a question that is this interface similar to the EGF receptor kinase dimer that Curian had reported? I think you said in the beginning that EGF stood out as the one lone different one, right?
Yeah, that's a great question. Yeah, it's totally different. The EGFRs don't require A-loop transphosphorylation, so those complexes are very different. And in those cases, one acts as sort of the activator kinase and the other as the receiver kinase. So the interfaces are very different, and the mode of activation is then very different. So EGFRs are probably the most studied system, but they're kind of the most abnormal, let's say, RTKs.
Mm-hmm. Okay. Yeah, given that we're 15 minutes after the hour, I think now the formal session is closed, and we invite you to come back next week. And thank you to both speakers. And now we have informal sessions, probably more NMR and EPR, magnetic resonance questions, technical questions. Yeah, you're welcome to stay.